Hello, everybody, and welcome to the ROSA Worldwide Webinar Series. My name is Mike Reed. I'm the Chief Scientist at ROSA, and I'd like to welcome you to today's webinar. To look for upcoming webinars in our series, please check the ROSA website or look for the announcements that periodically cross your email. During today's presentation, if you'd like to ask the speaker a question, you can press the Q&A button at the bottom of your screen, and we'll collect the questions for the speaker to answer at the end of the talk. Today's speaker is Maria Trujillo. She's a senior director at Merck. She's a seasoned quantitative pharmacologist with over 20 years of experience in drug discovery and development, leading enterprise-wide collaborations to address critical strategic and scientific questions through model-informed drug discovery and development. She's currently leading efforts in managing a group in the Department of Quantitative Pharmacology and Pharmacometrics, working in support of the MASH and obesity pipeline at Merck. Welcome, Maria.
Thank you, Mike. It is really a pleasure to join all of you for either a cup of coffee or lunch. I'm really excited to talk to you about some of the neat tools that we've been working with related to GLP-1 based therapeutic development. And my talk today, I want to focus on how we may accelerate the discovery and development of GLP-1 based therapeutics with MID3. So with that, I will move to the next slide. So I'd like to give you a little bit of perspective of the landscape of GLP-1 receptor agonists and their combinations in terms of therapeutics. And you see from the bullseye here, there are a variety of them at varying stages of development. This snapshot is from maybe about a year ago. And I can tell you from seeing one recently at ADA this weekend, it is intensely more populated than what you're seeing in front of you now. This field is growing in leaps and bounds with many, many new entries into the clinical development arena, as well as combinations of GLP-1 therapeutics with other assets and drugs. And the reason being, you can see from the right-hand side, is that there is quite amount of money to be made. In 2024, the estimates were about $53 billion. And I can say the number for that has gone up extensively as we've proceeded to 2026, where I think I saw the most recent estimate being somewhere on the order of $120 billion. This is still emerging as the market is changing. There are injectables at the forefront, many of which you are familiar with, such as Wegovy, Ozempic, Zepbound, Mounjaro, and many more are under development. Now the market has new entries that include oral GLP-1 based therapies, such as oral Wegovy and more recently Foundayo. These, instead of cannibalizing the injectable side, have actually expanded the market in ways not appreciated. And we are still learning just how big this pie is and how many slices may be divided up. So with that, it behooves us to explore how we may help our colleagues within our industry to accelerate the design and development of these molecules so that we may have a piece of that pie. I don't think I need to convince this audience too heavily about the value of model-informed drug discovery and development, and that many of you may be involved in modeling in your own way. But I'm sure you know that it can aid in dose selection, trial design, assessment of the competitive landscape. It facilitates decision-making. It can inform labeling and the use in special populations. And while I won't be able to cover all of these different topics in detail, I will highlight some and try my best to point you to some nice bits and pieces of the literature that you can begin to explore some of these through, and encourage you to keep digging. Some of the topics I wanted to cover today are different pieces or tools in my quantitative pharmacology toolbox. They include QSP, or Quantitative Systems Pharmacology, which is a mechanism-based approach that describes how the pathophysiology and pharmacology of molecules may combine in order to show weight loss. I also want to talk about physiologically based PK models, or PBPK models, which, contrary to QSP models, are actually showing you how the body will impact the PK of the molecules administered. And lastly, I want to talk a little bit about model-based meta-analysis, or MBMA. This is an excellent tool to provide assessment of external data from a variety of molecules in the competitive landscape, and can really help to condense and then interrogate these targets across clinical trials, providing learnings about the different molecules, the different targets, and aid in clinical trial design. These models all differ in the data that's used to build them, how they integrate knowledge, and the questions that they can address. And what I would also touch upon here, that I won't go into detail during this hour, but is that these molecules do not, these models do not need to be used in isolation, but rather I would encourage people to develop these models, use them in parallel, use them in tandem, and use them in ways that are creative to help join together the different ways and perspectives of using our data to inform drug development further. First, I'd like to start with QSP. There is a great QSP model available to study weight loss. This weight loss QSP model also has been modified to describe the effects of GLP-1 agonists on the reduction of food intake, body weight, and composition. This model was originally put together by Kevin Hall and colleagues and modified by those of Bosch et al. This model was developed using more than 50 studies that modified food intake and measured changes in body weight, body composition, total energy expenditure, and resting metabolic rate in humans. This is now used industry-wide to understand the impact of drugs on these different features and do so quantitatively. I want to show you by example how we can use this model in our studies. And this model can be used to recapitulate clinical data. And this was previously demonstrated by Bosch. Where you input PK of the molecule, potency, which can be in vitro, but could also be modified to be in vivo, and patient characteristics such as body weight, BMI, age, and sex. And through these inputs, you can simulate a clinical trial, such as the one shown below, which is an image of the step 1 trial of semaglutide 2.4 mg QW on body weight. And you can see on the right-hand side, the resultant simulations using this model fit very nicely with the observed data in dots. Not only can you look at weight loss, but you can also look at other features such as body compositions and the components of energy expenditure. And I'll show you more of those shortly. And here they are. The other beauty of this model is that it can be used to understand and simulate weight loss mechanisms that are beyond those of GLP-1 agonists, but also look at things that change energy expenditure and even fuel utilization. This can really be helpful in informing discovery and development of novel or next-gen obesity therapeutics. This example below shows how you can simulate semaglutide 2.4 mg in red. And then on top of that, which is the standard of care, you may also then look at what a drug or a putative drug that increases energy expenditure by 50, 100, 200, or 300 kilocalories a day, and see what that would do to body weight on the left, body
Maria,
composition. Yep.
Maria, are you on slide 7? It looks like on the screen we're seeing slide 7.
Oh, I'm on slide 8
Okay,
now.
maybe if you can, there you go. Thank you.
Okay, let me see if I, oh,
Okay.
sorry, I'm
Yeah,
going
now
too
I'm
far
seeing
ahead.
10.
8?
Yep, we're on 8. Thank you.
Okay, thank you, Mike. Right. So you can see here that on top of semaglutide, you can see what the effects of something reducing energy expenditure by a range of kilocalories per day might do to body weight, fat mass, and lean mass. So these features could be used to provide insights to novel drug therapies, because there are some drugs where we don't fully understand exactly how they're working. But so this can be used to better understand the mechanisms. This can be used to highlight mechanisms we may want to target and approach. And my favorite part, this can facilitate dose selection and trial design, especially for co-administration of multiple anti-obesity agents, where it gets to be quite tricky to see how some of these things may work independently, but others may work to synergize and give you greater weight loss than a single agent alone. And that level of complexity and pharmacology and physiology is best captured with a model like QSP. Now I want to talk a little bit about PBPK models. PBPK approaches are pretty common, and one of the uses may be to understand how the pathogenesis of obesity may affect the PK of drugs. Obesity is associated with physiological changes that may alter drug disposition, and some of them I have listed below, not in totality. There is some nice reviews available that I have listed. I've also noted that the FDA has recently called for comments in order to form a guidance for this in the future. So I know this is an active area of research, and it's seemingly not universal how these things may impact the PK of drugs. So more investigation is needed, but obesity is known to affect absorption by increasing gut permeability and gut flow. It may affect distribution of especially of lipophilic drugs, which may harbor themselves in adipose tissue and thereby affect the volume of distribution. It may affect metabolism and elimination by either altering the expression of related enzymes and transporters, or also increasing hepatic blood flow. So all of these features can be modeled a priori if you have the physiochemical properties for your molecule in question. They can then be tested against clinical data once you are able to look at the effect of obesity on the PK of your drug in early studies in the clinic. And then the PBPK model can also be of value in that it can be modified to aid in dose prediction of special populations within the obese participant realm, such as pediatrics and hepatic and renal impairment. So I think this is maybe the first stop for PBPK model, but I also wanted to share an example of how else PBPK modeling may be useful in the development of drugs for obesity. especially GLP-1 agonists. PBPK can help aid in the understanding of the effects of GLP-1 agonists on the PK of other drugs. GLP-1 agonists are known to delay gastric emptying, and this may impact the PK of other drugs. These effects are known to diminish with chronic treatment, but they must be assessed per health authority guidance.
Hey
Most
Maria,
often,
can you advance one slide? I think we're seeing both slides overlaying. 11. I think go to slide 11. There you go.
yeah.
Thank you.
See 11? Yeah.
Yep.
Okay. Thank you. Let me know if it, I see them perfectly attuned. So let me know if it,
Yep, will do.
if that's coming up. Sorry about this. So most often clinical studies are conducted with acetaminophen in order to quantify the effect of GLP-1 agonists on gastric emptying. And here is a really nice example using tirzepatide, which is a GLP-1 based agonist that is available on the market as Mounjaro or Zepbound. And you can see when acetaminophen is administered to participants on day -1 prior to treatment, you can view the blue PK profile. After tirzepatide's first dose is administered on day 2, you can see that the PK profile of acetaminophen becomes diminished. Both Cmax and AUC are lower, and Tmax is shifted. However, by day 23 of treatment with tirzepatide, these values do creep back up. Again, showing how the effect on gastric emptying, which is most prevalent at the initiation of treatment or upon uptitration of a new higher dose, diminishes over time once the participant has more experience with that dose and dose level. So this can be captured with a PBPK model. Prior to the use of PBPK, as I'll explain shortly, GLP-1 agonists went through a litany of DDI studies in the clinic in order to understand how this effect may impact the exposure or the PK of drugs of interest. Most of these drugs were believed to be sensitive to changes in gastric emptying, in other words, likely BCS class 1 type drugs, or drugs that have narrow therapeutic indexes. And this list of 10 drugs was frequently assessed in clinical DDI studies for some of the early GLP-1 receptor agonists, creating a large amount of work in the clin pharm space and slowing down development. The result of all of these clin pharm studies can be seen as an example in the label of Rybelsus, which is the oral semaglutide or oral GLP-1 agonist. And after performing all of those clinical pharmacology studies, the resultant label only lists concern over this effect, which needs to be where patients need to be aware that this may happen. And the only drug where there may be concern of this with happened to have been levothyroxine, where the exposure was increased. And as a result of that, the label suggests that there needs to be increased clinical or laboratory monitoring. So quite a bit of work for this label. Necessary at the time, but now with the use of PBPK modeling, we may be able to circumvent some of these DDI studies, if not eliminate them completely. An example of this is seen with tirzepatide. There, as I showed you earlier, tirzepatide, a study with tirzepatide in combination with acetaminophen was conducted in order to characterize the effect of tirzepatide on gastric emptying. This data was then captured with the PBPK model, and the effects of tirzepatide were well represented. Then the PBPK model of tirzepatide could be used to understand the impact of tirzepatide's delayed gastric emptying on other drugs. And this greatly diminished the need for doing many of those DDI studies and helped focus on where it might be most important. And the
Maria,
result,
can you advance one slide? Looks like it's overlaying.
yeah,
Here you go. Thanks.
thank you. I might have to just do that every time. Okay. Yes. So you see the resultant caution of use of oral medications when taking tirzepatide includes that of oral contraceptives, which was a DDI study that was actually conducted, but likely identified and facilitated by this PBPK modeling with tirzepatide, eliminating the need to do so many more studies. This will likely emerge as a method to aid in future development of agents, as the obese population is well known to take many drugs due to the prevalence of metabolic comorbidities. So use of PBPK can help understand the DDI potential with those a priori. And with the advent of oral small molecule GLP-1 therapeutics, it is very likely that many of these will be either co-administered or co-formulated, also requiring a good understanding of DDI potential, whereby PBPK modeling can give you a preview to issues should they arise, and or understanding of many of the potential interactions to be had. All right. Let's move on to model-based meta-analyses. Model-based meta-analyses can characterize weight loss that is observed in clinical trials with obesity assets. And this is a great way to amass and understand a lot of the data that is coming out. As I showed you earlier, there are many, many, many molecules under development right now in terms of obesity therapeutics of different routes of administration, modalities, and target combinations. This tool can really help you aggregate and understand the available data in a way that is digestible. It can also help you integrate data across these clinical trials that vary in not only treatment, but also design and patient population to help you draw insights about this competitive landscape and inform and accelerate drug development. And I'd like to show you a few examples of how that may happen. Here's an example of some of the data that's been collected.
Advance one slide, please.
Yeah.
There we go.
Okay. Here's an example of the data that's been collected from a variety of obesity trials using GLP-1 therapeutics. This slide is from a couple of years ago, and I can tell you now it's even more enriched. But you can see how there's a plethora of longitudinal data across a variety of molecules. So there's a lot that we can learn from here. In addition to weight loss, these data sets can be designed to include additional secondary endpoints like HbA1c, lipids, blood pressure, heart rate, gastrointestinal AEs, and more. So learning from this source is immense. And it's really about how you tackle using these data that will determine your ability to learn different elements from it. But I highly recommend this methodology because there is so much potential. Here is an example of some learnings you may take away from just cutting up the data in different ways. This is a depiction of some of the GLP-1 agonists that are just pure GLP-1, and they are either administered orally or subcutaneously. And this data shows that weight loss, whether it is administered by either route, can give you the same potential level of weight loss, such that it doesn't matter how you get it in there, as long as you are able to achieve concentrations likely that get that then you will be able to achieve the weight loss that is desired. So route of administration should not impede the level of weight loss. In these data sets, not only do they have the weight loss data,
Advance one more, please.
the low weight
There you go.
loss data over time, but they also include all the details about every arm in a trial, how it's titrated. You can see that on the left. The patient population characteristics of the trial. Maybe they're diabetic, maybe they're non-diabetic. There can be other features examined as well. And then it also feeds on the concept that early weight loss predicts late weight loss. And this has been seen across mechanisms with drugs that decrease body weight through food intake and may extend to other mechanisms as well. And it's these different factors that can be combined quantitatively to build a model for each of the drugs that you have weight loss data for, provided you have sufficient longitudinal data. If you're able to build this model, you can then predict how your molecule of interest may behave, not only in the trial similar to what has been conducted, but you can predict longer term weight loss, and you can predict weight loss in different patient populations using different titration regimens, and really can be helpful with clinical trial design. Here's an example of how some of these molecules stack in terms of their predictions versus their observed data. And you can see that this model does perform very well across different molecules and across different patient populations. As long as the perturbation is within the observed data and not extrapolating, with the exception of long-term versus short-term weight loss, this method of modeling can be extremely useful in comparing assets and designing clinical trials. Here is an example of how you might go about doing this, where you may want to compare your drug A or B, to semaglutide sub-Q or tirzepatide. And you're able to predict all these drugs in the same clinical setting in a similar trial design, and compare them head to head and understand where you may fall compared to some of these drugs that are already on the market, and help you understand whether your trial in its length, duration, and treatment regimen may enable you to differentiate, or whether your drug has a high POS of meeting or exceeding the bar set by semaglutide or tirzepatide. Again, a very powerful tool for clinical trial design and probability of success in reaching weight loss goals. Okay. Now I want to talk to you a little bit about co-development of anti-obesity agents. Co-development is tricky. There is regulatory guidance for combination therapies, but it is not as simple as it sounds. Combination products can be not only two different assets combined, but can be products and devices and a variety of other things. There have been combination therapies for obesity that are currently on the market. Tirzepatide is one of them. There are others such as amycretin, survodutide, etc. Many of these combination therapies that are ahead in terms of their clinical development or place in the market have been unimolecular. And they don't need to follow the guidance for co-development that I want to discuss with you in greater detail. It's a little bit more streamlined when it's unimolecular and less complex. But now that we are getting into combination therapy in the sense of different assets, either co-administered or given as fixed dose combination products, this is where I really believe MID3 can do a lot to help us to accelerate the process. But first, I want to share a little bit about the guidance so you can appreciate what the burden is and maybe imagine how you can use some of the tools I just discussed to prosecute. Some drugs that are already far on this path include cardiometabolic, and there are a variety of small molecules that I mentioned earlier that are on the market that will likely explore fixed dose combinations in the future. So be on the lookout. All right. So for co-development, molecules that will go for co-development need to meet a specific criteria. The combination needs to be intended to treat a serious disease or condition of which obesity is considered. There needs to be a strong biological rationale for the use of the combination, and there needs to be a full non-clinical characterization of the activity of both the combination and the individual new investigational drugs, or possibly even a short-term clinical study to establish the validity of the combination, that in order to suggest that the combination may provide significant therapeutic advance over what is currently available as therapy, and that it's superior to the individual agents. That's a lot of work. And you also need to show that there is a compelling reason why new investigational drugs can't be developed independently. So this puts quite a bit of burden on the development process. So maybe let me break it down a little bit further. So there are requirements that apply to our development process for two or more molecules, both individually and in combination. So for clinical pharmacology, that means the main objective of the early studies are not only to characterize the pharmacology, the pharmacokinetics and safety of individual assets, but also the combination. So that's not one study, that's three. And that you also need to provide data to support appropriate dosing for the combination in a phase 2 setting. So quite a hefty load. Once you do get into late stage development, you need to further demonstrate the contribution of each individual and combination to the extent possible. And you need to provide evidence of the effectiveness of the combination. Lastly, you're going to have to optimize the dose or doses of the combination. So these factorial studies can become enormous. So this is where I would like you guys to entertain how these quantitative tools we've discussed may accelerate the co-development of these anti-obesity agents. How can we use this quantitative toolbox to provide more efficient discovery and clinical development paths? For QSP, I believe it can facilitate decision making and reduce our uncertainty, both in target selection, early trial design, and dose optimization. It might not provide all of the answers, but prior to having a solid understanding of how each asset performs in the clinic, QSP can get you there, or at least narrow the focus to something that is amenable to study in these clinical trials. PBPK can be used to streamline or even circumvent some of the clinical drug-drug interaction studies that I had mentioned previously with the example of tirzepatide. And lastly, MBMA may aid in go, no-go decisions, as we call them in industry, based on the assessment of the probability of success of this combination versus other molecules that are out there in the competitive landscape. Some of the combinations being entertained in obesity are not just dual agonists, but even triple and quadruple agonists at this time. It's becoming increasingly competitive. And being at the ADA meeting this weekend, there was new data that came out on retatrutide showing that it has some of the best weight loss to be seen. That is a triple agonist at this point. So keeping abreast of the competition is extremely important, especially given the expense of drug development and the need to succeed for each of our companies. This MBMA can really help you make decisions and allow for the appropriate allocation of resources and the success of our efforts. So with that, I'd like to pause and thank everybody for your attention and give special thanks to some of my colleagues for helping with some of the slides, and especially to my team at Merck, who I really enjoy working with on all of these types of models and more. And with that, I'm happy to take any questions.
Great. Thank you, Maria. Excellent talk. So I do have a couple of questions here. One is, as you've mentioned, this is a very crowded field. And so differentiating a new approach or a new agonist or combination of agonists is challenging, especially if you're looking at lengthy weight loss studies. Can you speculate a little bit for how you would apply these different approaches to do competitive differentiation to figure out whether you do have a spot in the market that's unique or advantageous?
Yes, excellent question. Probably a very complicated answer, as this is emerging. Up until recently, I think weight loss was something that was still pushed. How can we get more weight loss with any of these assets, right? So that's why you're seeing the double and now triple agonists, and in fact, there's only a handful of assets out there, not yet approved, that do achieve 25 or so % weight loss, but it is happening. So people are questioning whether the amount of weight loss alone is going to get you there, or whether you need to differentiate in additional ways. And it's unclear how patients will drive this. Some may want to lose that much weight because that's basically bariatric surgery like weight loss, which is amazing. Who would have thought 5 or more years ago that we would ever be here? I don't think any of us did. This is happening so rapidly, and the numbers were just going up and up and up with all of these injectables. But we also appreciate that injectables are not for everybody. They don't travel well, to say the least, and they can sometimes have higher prices.
Hmm.
And now we're trying to make some of these oral versions, and most of the orals at this point in time have been around 10% weight loss. So pushing the orals and finding ways of, getting the orals up to the level of weight loss we see with injectables is something that I think some of these tools can be used for. Um, and in particular QSP, where you really like need some of the pharmacology to help understand like the potential of your drug versus some of the injectables and go um, cross modality to understand that better. Um, and I also think that if we can develop MBMA or some other tools to better understand the GI tolerability of the different assets, that is another driving force in not only uptake of patients on these, because GI tolerability is the major complaint, I'll say. If we're able to find a way to differentiate on tolerability, of which to this point has not really happened, because most of these drugs, though they may vary in the level of weight loss, have been hovering around similar numbers in terms of GIAEs.
Hmm.
That will also be an area to differentiate in. But as of now, a lot of you know things are moving very quickly, so figuring out how to differentiate in those areas is challenging. So I could probably talk about this for about another hour or two.
That's
I do want
okay.
to. I hope that gives people some reasonable uh, answers.
Thank you. Next question is, can you speak to the use of MIDD in the development of antibody drug conjugates? Are there models available that could be used for this class of drugs? There are unique
Antibody
aspects of
drug.
ADCs that might be uh, applicable to some of the models that are out
So I haven't seen any ADCs come into the obesity sphere to date.
there. Hmm.
I got to say, I'm not as familiar with ADCs. So I'm going to have to pass on that for now, but I would like to answer that at some point for myself.
Yeah, I think of these more in the oncology space, perhaps, but
That's great.
there are people have done modeling. This is for the questioner. People have done modeling in this area, and perhaps you can look in the immuno-oncology space to get some
That's right.
examples there.
That's
Okay,
right.
next question. Could you please comment on using GLP-1 to treat MASH? Have there been any QSP models developed to evaluate this?
Yes, I believe NAFLD Sim. I think I actually published a paper on this with some of my colleagues a couple of years ago. And you can use NAFLD Sim to represent GLP-1 agonists very nicely. And that's what I would recommend.
Hmm.
Um, yeah.
Great. Okay, now a really speculative question. This one's from me. Can you talk a little bit about the role of artificial intelligence in not just drug discovery in general, but maybe focused on the development of modeling tools or the integration with some of the modeling tools you talked about, and where you
Mhm.
see this going in the future?
That's an excellent question. I think that's a question on a lot of our minds. I was talking with some folks on ISIG for this just the other day, because I think a lot of us are asking this, but I don't know that anybody has like the perfect answer. So mine
Hmm.
will likely be imperfect. I think I have some experience using AI ML tools with some of my colleagues here at Merck for a variety of purposes, and some of them are revolving around these GLP-1 therapeutics, as well as MASH. I think there are ways that you can apply those where it is a standalone to maybe address questions where we really don't understand how things are coming together mechanistically, and we may not this have an explanation that revolves around exposure response per se, right?
Hmm.
Because many of these AI tools, you don't know why, but there's an association and it can model this. It's just
Yeah.
modeling what it seems, right?
Hmm.
But I think that there can be very valuable applications of this in that way. And I also am very interested to explore how we can better use some of these AI tools to maybe refine some of the more complex methodology that we have, such as QSP. And I know there are many, many ideas about how to do this. Um, and I and, and I'm very happy that people are very enthusiastic and excited in testing these applications. I believe I reviewed like many abstracts about this going into ACOP this year. So I see a high volume of interest in this area, but I don't know that there's any specific way. I think you can use it to maybe simplify your model. For example, I've
Hmm.
seen examples where, like, once you get a really nice QSP model, maybe you can use AI ML to sort of supplant what it is doing and
Hmm.
accelerate some of your run times. Because some of these models are, the model here that I mentioned today is not so bad. The run times are not so terrible, but some of the more complex models in the metabolic field can take a lot of computing power and be very difficult in terms of the
Hmm.
runtime. So
Yeah.
maybe AI/ML can help accelerate that runtime by sort of being like an overlay.
Hmm. Yep.
That's one way, and there can certainly be um, I'll say places in between where you
Yeah.
can leverage some of these AI tools to help bridge a gap, maybe in understanding where you might not have data and you need to make some connections. I know I'm speaking of this in great vagaries because I am not an expert in AI ML, but I'm very lucky to have excellent colleagues here at Merck who are.
Hmm.
And this is just my very dumbed down perspective of how to apply these things. But I think there's a lot we can do there. It's just a matter of finding ways that are actually helpful and
Yeah.
useful. Because I feel like it's often now we have a hammer and everything's a nail,
Yep.
and I think we're in that space. But I think until we start to, it's a matter of trying and exploring, and when things don't work or things are not worthwhile, then we're learning, right? And I think
Hmm.
that can be said of any AI tools. We're all guinea pigs at this point, and over time, I think it will become more streamlined and will understand where these tools are most helpful rather than just using it all the time everywhere for everything.
Thank you. Two more questions. First is, how is MID3 being applied to model and predict tolerability for
Mm.
GLP-1s? And are there currently models capable of doing this, for example, integrated within QSP frameworks?
So I am aware of a model that integrates this within QSP, and that's Obesity Sim. I am also aware of most big pharma has been using Markov chain modeling to get an understanding between dose exposure and GI AEs. That can be quite complicated, but that does provide great insight if you have the data. Um, and I think that that's an area like that is one of the harder areas I would say to understand and explore, whether you have individual data or you have data from competitors. It is often not reported very consistently, and it is arduous to collect such data with great accuracy in clinical trials, but people are.
Hmm.
And it's quite variable. Um, and I'm not clear how well clinical trial data and real world data will mesh. So I
Okay.
think that there can be many attempts at doing this, and it will be interesting to see how all of these things line up and how we may simplify streamline titration regimens, because currently the standard is dose doublings for 20 weeks to get to a
Hmm.
top dose, and that is a long time.
Yeah.
Obviously people don't like waiting that long, and many people don't make it to the top dose,
Yeah.
um, at least in the real world. So how can we get people there faster, but also some people don't want to go through what people have to go through to get there.
Yeah.
And I wonder if some more novel mechanisms or better understanding of the biology that causes some of these GIAEs may be needed to truly make a difference here.
Thank you. Um, Next question. How reliable have these simulation results been? So specifically, have you or has it been reported post-hoc analyses to see if the decisions based on MID approaches were correct, or maybe I should say useful?
Useful, yes. So I think some of these models, like the MBMA model, has performed very well. It's still too early to say whether some of these QSP approaches can predict as can predict well. But I'm very eager to see that with some of the newer molecules coming out, and especially novel mechanisms. So I think that is a TBD answer for
Hmm.
the most part. Um, I do see a lot of use of PBPK, as I shared, and I do see the MBMA is doing well, because that the data that fuel those comes out very quickly. Um, the QSP model, I think we're still waiting on the results.
Okay. I do have one more question. Which are there generic open source tools out there that are useful for QSP model building? And
Mm.
if you can help point this questioner just to a resource where they can maybe find these tools.
Huh. That is a great question.
You
I
know,
wish
there are
I had,
Git,
maybe
yeah,
you
there
know.
are GitHub. Yeah, I think there are GitHub um, folders where you can find models that people have built that are open source and publicly available. And honestly, just do a Google search on these. There are model repositories too that are open source and available. These days, a lot of publications actually have to submit their model files.
Hmm.
And so you can often access those model files and run them. And as far as open source software, I know a lot of academic investigators use R as an open source um, software platform. But there are certainly other commercial modeling softwares out there that are not that expensive that can be used as well. But maybe that would be a place to start to look for these.
Yeah, perfect answer, Mike.
Oh, thank you. With that, I would like to thank our speaker, Maria Trujillo, for joining us and giving us an excellent presentation today. Just to remind the audience to please check the ROSA website for upcoming webinars. And I'd like to thank you for your attention today.
All right. Thank you
Great.
all so much.
Thanks, everybody. Bye-bye.